I'll try to get these lectures up as soon as possible. So I know this is a lot of material. I was hit by a car a couple weeks ago, or my car was rear-ended, and my computer, in addition to my car, got totaled. So my ability to edit and get videos up is a little altered right now, and sometimes the phone will directly post and sometimes it won't. It's just kind of a serendipity if it does happen. Okay, so now what I want to do is I want to move on and I want to talk about these neurotransmitters. So these neurotransmitters are chemical messengers that are released by neurons. And again, they can either excite or inhibit the tissue that they're acting on. Okay. And just some terminology before we get too deep into this. Presynaptic, when I say presynaptic, I'm talking about the neuron before the synapse in space. Or postsynaptic is going to be the effector, what's getting acted on. Okay. So when I say presynaptic, I'm talking about before the synapse. Postsynaptic is going to be that effector, what's getting acted on by the neurotransmitter. So these neurotransmitters are going to interact with receptors, okay, or what we call chemical receptors, because there are different types of receptors, but these are chemical receptors. There are two general types of chemical receptors. The first type is a little easier to understand, and this is known as the ionotropic. An ionotropic receptor is an ion channel. This is a ligand-gated ion channel, like a sodium channel that opens when a neurotransmitter bonds to it. A neurotransmitter binds to it, it opens up, and ions flow through those channels. Does that kind of make sense? So it's pretty straightforward. Ionotropic receptors are pretty straightforward. When a certain neurotransmitter attaches to them, they'll either open or close. They'll either allow ions to flow in, or they'll close and ions won't flow into the neuron. Make sense there? More or less? Okay. The other major category of chemical receptors are what are known as the metabotropic receptors. These are a little more complicated than the ionotropic. And when I think of metabotropic, I just think of metabolism. So lots of stuff goes on. Okay. The big thing that you want to throw in the back of your mind is that metabotropic receptors involve secondary messenger molecules. So another molecule is produced that then alters processes that go on within the cell. That's known as a secondary messenger. The typical thing that happens with these metabotropic receptors, this is the classic thing that happens with these. So here I have my receptor, okay, and I'll have a neurotransmitter is released, okay, and that neurotransmitter will then attach to the receptor. It will induce a conformational change. Now, instead of just an ion channel opening or closing, something more complicated is going to happen. What's going to happen is that receptor is generally associated with something known as a G protein. Okay, a G protein. And this is a protein on the inside of the cell that has several subunits. And what happens is when that receptor undergoes a conformational change, that conformational change is transmitted to the G protein, and that causes the G protein to kind of break up into different subunits. Okay? And then those subunits act on an enzyme. Okay? And typically the enzyme is something known as adenyl cyclase. Adenyl cyclase. And then what adenyl cyclase does is it is an enzyme that catalyzes the production of a secondary messenger molecule inside of the cell. That secondary messenger molecule is created, and then it goes on to alter activities occurring within the cell. Okay? Does that kind of make sense there? That's why this is metabotropic. It involves metabolism. There are lots of moving parts. So it's a receptor, it's a G protein, adenocyclase, secondary messenger. So these are often also known as G protein coupled receptors. Okay? G protein coupled receptors are metabotropic receptors. These are the two major chemical receptor types.
They're G protein what receptor?
Coupled.
Coupled?
They're coupled or attached to G proteins on the inner inside of the cell or on the inner surface of the cell membrane. So far so good? Sort of. All right. So let's talk about the general types of neurotransmitters. Okay, this is going to be kind of general for now. Okay. So we have three general types that I want to talk about. First of all, the one thing that you need to understand is neurotransmitters will excite or inhibit, but it's not the neurotransmitter necessarily, it's the receptor for the neurotransmitter. So it's going to be very tough for me to say that a certain neurotransmitter is always excitatory or always inhibitory. Because that neurotransmitter, depending on the receptor that it attaches to, will be inhibitory or excitatory. And some receptors for some neurotransmitters inhibit and some excite. I'm going to give you some general rules of thumb, though. Okay, some general rules of thumb, but rules of thumb are not always cut in stone, right? There's always some nuance there. Okay, so you have the amino acid neurotransmitters. Remember what amino acids are, right? Amino acid is NH2, alpha carbon, an R group, and a carboxyl group, right? That's an amino acid there, okay? The major amino acid neurotransmitters are glutamate, aspartate, D-serine, so it's the D-enantiomer of serine, something known as GABA or gamma aminobutyric acid, hence why we call it GABA. GABA or gamma aminobutyric acid and glycine. Okay? These are the 5 major amino acid neurotransmitters we run into. I want you guys to definitely know glutamate. Glutamate, okay, is the most common neurotransmitter in the central nervous system, and it is the primary excitatory neurotransmitter. Okay? It is primarily excitatory. All right. So when glutamate attaches to glutamate receptors, that tends to excite other neurons. That tends to make other neurons more prone to depolarizing. Okay, so that increases activity within your central nervous system if you have a lot of glutamate. Okay. The next one is diametrically opposed to glutamate, and that is the gamma aminobutyric acid, or GABA. GABA is the major inhibitory neurotransmitter in the central nervous system. Okay, so this makes neurons less prone to depolarize. This one is really important, specifically the receptor for GABA, which is known as the GABA receptor, and there are actually 2 types. There's the GABA A and the GABA B receptor subtypes. But what happens here is when GABA binds to a GABA receptor, typically what we see happen is the GABA receptor opens up, and as you might imagine, if we're talking about ions, GABA receptors are ionotropic, aren't they? inotropic receptors. Okay. So GABA binds to the receptor opens up, and that allows chloride ions to flow into the neuron. Okay, so inside. What does that do to the neuron though? So GABA bonds to the receptor opens, chloride rushes into the neuron. What does that chloride do to the membrane potential? Does it make it more or less negative? It what?
Hyperpolarized?
There you go. It hyperpolarizes. Okay? It makes it more negative, right? The resting membrane potential, okay, and here's a graph. So your resting potential's right here, right? And so I have to get up here to get to that threshold to cause it to depolarize, right? That's this graph here. Now in this graph, I have increased levels of GABA. Increased levels of GABA. And so what has happened is the membrane potential has become more negative. What does that mean? Now that it's more negative, what does that mean?
It needs even more positive energy to reach its actual
So
potential.
I need to have more excitatory input, right? I need much more excitatory input before that neuron will depolarize. Because right here, I just need this much, right? I need that much in my graded potentials, right? Cause that much sodium to shift in to get the threshold to trigger that thing off. But in this case, I'm going to have to stimulate that neuron much more to get it to depolarize. So the neuron has been suppressed, right? It is suppressed, it is inhibited due to the hyperpolarization. The reason that I'm talking about this specifically is there are many drugs out there that act on the GABA receptor. And these drugs are very important because they are used to treat things like seizure disorder. If somebody's having a seizure, that's often due to random chaotic firing of neurons, right? The neurons are depolarizing everywhere in your brain, specifically in your motor cortex, and you have that seizure activity. So if I give somebody a drug that interacts with these GABA receptors and helps allow more chloride into those neurons become hyperpolarized, and are they more or less likely to fire? They're less likely to fire, so they're suppressed, and the seizure activity is going to decrease. Does that kind of make sense? Mm-hmm. Now, unfortunately, these drugs tend to decrease neuron activity everywhere. So what else do they do to our patients if we give them these drugs?
They can lose movement.
What else might you expect? How might they act?
Loopy.
Yeah, they're going to be kind of tired, loopy, and they're going to be overall depressed. Not as in depression, but overall you're going to see depression, right? So they may be very tired, and if you give them too much of these drugs, they may even quit breathing on you if you suppress their central nervous system enough. And the two major classes of drugs that work on GABA receptors are what are known as the benzodiazepines and the barbiturates. And benzodiazepines are really common. Right? These include drugs like Valium. You guys have heard of Valium, right? Sometimes people take Valium for anxiety and for sleeping as well, and sometimes for muscle spasms. Also for seizures, right? So Valium, Xanax. Are you guys familiar with a drug called Xanax? It's a common sleeping drug. Alprazolam, well, which is generic Xanax. Sometimes it's given for anxiety. Okay. Again, so Valium, Xanax, Ativan is another one, or lorazepam. These are all benzodiazepines. Another class of drugs, which is older than the benzodiazepines, are what are known as the barbiturates, and the classic one is something called phenobarbital. And phenobarbital was one of the first drugs that we created that could actually treat seizures. It's not as commonly used now, because the barbiturates tend to be more potent than the benzos. The barbiturates can directly cause these GABA receptors to open up at high enough doses. The benzodiazepines don't directly cause the receptors to open up, but what they do is when the receptors do open up, they allow more chloride in than normal. So they enhance the activity of those chloride channels. Yeah.
Does tramadol fit into those sections at all?
Tramadol, I believe, is an opioid.
I thought.
Yes, it's not. It doesn't work on GABA receptors. It does suppress the nervous system, but it works on other receptors. We'll actually going to talk about that here in just a few minutes. But tramadol is it's not an opioid chemically, but its actions, it acts on some of the same receptors that opioids act on. And because it doesn't chemically fit into that kind of opioid thing, tramadol is actually not considered a scheduled drug like Vicodin and some of the more classic opioids. So you can go to like Mexico, right? You can go to Mexico and buy tramadol over the counter. You can't necessarily do that very easily with, like OxyContin or something like that. But yeah, tramadol does, all of the opioids actually cause suppression, for sure. Okay.
So, what kind of medicines do they use typically now for people with like epilepsy and things like that?
They may use these. There are other medications that have some really interesting and unknown effects. In some cases, so things like phenytoin, Cervex, phosphonatoin. Those tend to be some of the newer anti-epileptics that work in strange and unusual ways.
So probably the biggest problem we have with epilepsy is we still don't really know exactly what is causing it.
We don't have like we don't know exactly. Okay, moving on to the next major category of neurotransmitters. These are the biogenic amines. So these are like the amino acids in that they have an amine group in them, okay, but they are not acidic. They do not have a carboxylic acid group in them. Okay, and these are five major ones. Dopamine is a big one. Norepinephrine, epinephrine, histamine, and serotonin. Another name for serotonin, it commonly goes by its chemical name, which is 5-hydroxytryptamine, or 5-HT. So if you see 5-HT or 5-hydroxytryptamine, that just means serotonin. That's the chemical name for serotonin. You guys okay with that? Yeah.
You mean, my mom takes some pills for stress, 5-HTP, they're natural, is that what it is?
That's like a supplement kind of thing, right?
Yes,
5-HTP.
feel happy or
Yeah, it is a it's not serotonin, but it is a molecule that might be able to be metabolized into serotonin.
But it does not.
Huh? But that no.
Okay.
Yeah, this is 5-hydroxytryptamine, 5-HT.
Oh.
Yeah.
But you can take serotonin, right? Or they're like.
Well, yeah, you can take a lot of serotonin, but the problem is, remember these biogenic amines tend to be kind of charged, right? Because they have nitrogen in them, and charged stuff tends tend to get through the blood-brain barrier very well. So a lot of these supplement your neurotransmitters.
Would it be
Doesn't
effective
really,
for
no.
somebody with depression or with
Really.
low levels?
I'm
is it?
aware of, no, because again, you can take it, right? You can swallow it, you can inject it, but is it going to cross the blood-brain barrier? That's the question you have to ask. And most of these won't, right? Like I said, you can inject dopamine, you can inject norepinephrine, you can inject epinephrine, and these tend not to cross the blood-brain barriers. You can't get those into the brain. Okay, so with that in mind, I do want you guys to focus on particularly dopamine and serotonin at this point. Okay? Dopamine is a very important neurotransmitter when it comes to mood, when it comes to movement, and when it comes to reward pathways in your brain. Okay, so dopamine is really important when it comes to problems like Parkinson's disease. We talked about that. One of the problems with Parkinson's disease is you have a loss of dopamine-producing neurons in an area of the brain. It's actually an area called the substantia nigra of the brain. And you end up losing your ability to have fine motor coordination. And so what happens? These people tend to get tremors, right? They don't have fine motor coordination. They tend to have what's known as an ataxic gait. They can't walk. They have poor balance, and they can even go on to develop dementia and other problems. Dopamine seems to play a very important role in addiction, in the reward pathways of our brain. When we do something that is rewarding, okay, dopamine tends to be released by neurons, okay, and this is somehow creating that good feeling that we associate with certain actions, certain activities, and even the use of certain substances. And so dopamine looks to play a very big role, chemically at least, in addiction and developing dependence to a whole bunch of medications and drugs, opioids and alcohol as well, when these reward pathways get activated. And I just remember there's The Simpsons, and Homer's brain is talking to him, and it's telling him, go ahead and eat that donut, Homer. And what happens when he eats the donut? The brain's like, here you go, Homer, here's some dopamine. He goes, ah. Now there's another example of this. It is a cartoon called South Park. I don't know if you guys are aware of that or not. Unfortunately, I can't really talk about this in any detail, but it's very kind of a crude. But basically, one of the boys has a problem with playing video games, and Satan visits him at night and explains the biological basis of addiction with dopamine and all that. So now you can go to YouTube and watch it and go, oh yeah, okay, it makes sense. Yeah, it's a neurotransmitter. Yeah, but it is involved in pleasure and reward centers. Dopamine also tends to be, or looks to be involved in psychotic behavior or psychosis. And psychosis just means a loss of contact with reality. And so people that have diseases like schizophrenia, for example, what we do is if we give them medications that block dopamine blocking medications, their psychotic symptoms go away. And so dopamine looks to play a very important role when it comes to psychosis, and having too much dopamine in certain areas of the brain may be associated with psychosis. So it's an important one. Serotonin is another very important one, particularly when it comes to mood. Okay, and of course there are lots of medications that we've developed that aim to enhance mood, or what we call antidepressants. And the most common category of antidepressants are known as the SSRIs. And that stands for selective serotonin reuptake inhibitor. And one of the major mechanisms by which we get rid of neurotransmitters is through reuptake. So there is a pump that pumps the neurotransmitter back into the neuron, or sometimes it gets pumped back into like an astrocyte and gets processed.
Back up the ion channel.
Huh? No, it has its own channel.
Okay.
Yeah, it has a it has a reuptake channel. And what we've hypothesized is that some people, if they don't have enough serotonin in certain areas of their brain, they may have mood alterations of mood. They may develop dysthymia or depression. And so these SSRIs inhibit those receptors that reuptake. And so if you inhibit those, that means that the serotonin doesn't get reabsorbed, and it stays out in the synapse longer, and it can have an enhanced action. So you, in essence, have more serotonin available. And so if you increase the serotonin levels, that in some patients may enhance their mood and help treat their depression. So these are things like Prozac, Paxil, Celexa, Wellbutrin, if anyone has ever, or bupropion, anyone ever taken that to help them quit smoking. But serotonin also has an important role, we think, in psychosis, just like dopamine. And there are medications that block serotonin, and they, and some patients are very effective at treating the psychosis. And we also know that drugs that can alter our sensorium, okay, can alter our sense of reality. Drugs like LSD, psilocybin, magic mushrooms, mescaline and peyote. These kinds of drugs that can produce hallucinations tend to activate serotonin neurons, tend to cause serotonin neurons to release their serotonin onto what are known as the 5-HT2A receptors, or the serotonin 2A receptors.
Is that the drug 5-HT?
No, that's a receptor for serotonin. So when I talk about serotonin, the problem is I've super simplified this. But with serotonin, there are at least 7 receptor subtypes for serotonin. There's 5-HT1, 5-HT2, 5-HT3, 4, 5, 6, and 7. And then each of those have different subtypes. For example, the 5-HT2 have the 5-HT2A, 5-HT2B. Okay, so there are a dozen or more receptors for just serotonin alone. And specifically, it is the 5-HT2A receptor that seems to be a major part of how a lot of these drugs, these hallucinogenic drugs work, LSD, psilocybin, DMT. And in fact, most of these drugs are chemically almost identical to serotonin. Like DMT, dimethyltryptamine, is chemically almost serotonin.
I thought DMT would help dopamine.
Huh?
I thought DMT would help dopamine. It produces more dopamine.
No, it primarily, this is, again, there's a lot of nuance here, but primarily a big part of it is the 5-HT, the serotonin. And if you look at DMT and you compare it chemically to serotonin, they're so similar. Yeah, they're so similar. Yeah.
We already have, don't we release DMT when we pass away
Uh,
or something like that?
They say that we release it like 3 times within.
I don't know that that's been validated with studies though. I'm aware that we have, the research that we've done is we've taken DMT and we've put it in vesicles of neurons, and then we've stimulated the neurons, and when the neurons depolarized, the DMT was released by the vesicles. So it is plausible that DMT is a neurotransmitter. And I think looking at rats and mice, They've been able to see that there's DMT in the pineal glands of these animals. So, you know,
It's possible.
yeah, it's plausible. There's plausibility there.
It's like 3 times within a lifetime.
What's that?
They say it's released, we release it 3 times within a lifetime.
Yeah, I don't know.
Yeah, they say.
Yeah.
I don't know how true that is,
I don't know, but yeah, these are highly serotonergic. Now, other drugs like mescaline, for example, mescaline has and ecstasy as well, it has a very strong dopamine component to them, and of course serotonin as well. So it's not all or nothing. It's a pretty complicated mix of things going on there. But yeah. So again, take-home point, these are very important when it comes to mood, when it comes to movement, when it comes to how we interpret the environment. Okay, time. And then the last neurotransmitter I want to talk about is acetylcholine. We've talked about this already with the muscular system, right? This is the most important preganglionic neurotransmitter in the autonomic here. And it is the most important neurotransmitter when it comes to the somatic nervous system. So, I had already mentioned it, but how do we get rid of neurotransmitters? Well, there are basically 2 ways. We degrade them with enzymes, or we reuptake them. We transport them back into the neuron or the glial cells. And in some cases, we do both, enzymatic degrading and reuptake. For example, acetylcholine, what is the enzyme? What is the major enzyme that helps break acetylcholine down? You mentioned this in the neuromuscular junction. It's the acetylcholinesterase. So acetylcholinesterase breaks acetylcholine down into acetic acid or acetate and choline. And then the choline part gets sucked back into the neuron, gets transported back into the neuron, okay? And then acetyl-CoA that comes out of the Krebs cycle attaches that, okay, acetyl group back to the choline, and now you have acetylcholine again. So, there you go. Now, occasionally, to make it even more complicated, right? More complicated than it already is, occasionally, nerves will directly attach to other nerves or other tissues without there being a synapse. So they'll directly attach, and there'll just be a gap junction there. And in that case, the wave of depolarization can be directly communicated into that neuron or that tissue, so you don't need to release neurotransmitters in that case if it's directly attached, right? And we call these, okay, electrical synapses, okay, not chemical synapses, okay, because here you have a space that a chemical needs to kind of cross, but with an electrical synapse, it's a direct connection. This tends to be a very specialized structure. In the classic area where we see this would be like vision, okay, where you have a direct attachment. You do not have that chemical synapse occurring there.
So it's not very common.
Not as common, yeah. It's not the classic we talk about. Okay, moving right along. We only have a few more things to talk about, and we should be able to wrap things up here. We won't be out as early as some days, but I think we'll be able to get out on time. So the next thing I want to talk about are the general types of nerve fibers. And there are 3 general types, and they're classified by how fast they can transport an impulse, how large they are, and whether or not they have myelin. And these are A, B, and C. Okay. So the type A nerve fibers are myelinated. They're myelinated, they tend to be large, and they tend to transport signals very quickly. Approximately 140 meters per second. So what is that in feet per second? Anyone care to guess? Well, what's a meter? What's a conversion from meter to feet? a little over 3 feet. 3 feet and some change. So 1 meter is about 3 feet and some change. So that you're looking at approximately 400 feet per second. Right? So a little over a football field in a second. That's how quickly those impulses are moving through your body. So as you might imagine, these types of nerve fibers are going to be really important when it comes to touch, right? It comes to touch and motor impulses, right? Because you want quick, rapid actions. Right? Your type B nerve fibers are also myelinated, but they are of intermediate diameter. They don't have as large a diameter, and so their rate of speed, their conduction, is going to be slower, around 18 meters per second. Okay, so that's 70 feet per second or so. Okay, and these tend to be sensory and some peripheral sensory information. Okay, it comes from peripheral. And then finally, you have the unmyelinated fibers, and as you might imagine, not going to be very fast at all. You're only looking at about 1 meter per second. Okay, and this tends to be involuntary muscle and glands. And so you would see this in like the smooth muscle of your lungs, the smooth muscle of maybe your intestines, mucus secreting glands and things like that. So there's going to be a significant delay in that information getting to those and acting upon those tissues there. It's not going to be nearly as fast as your type A nerve fibers. All right. So far so good? So what I want to do now is I want to move right along into what's known as inhibition and facilitation. So to make this even more complicated, not only can neurotransmitters work here, work on the dendrites, right? You could have dozens of neurotransmitters acting upon a neuron at one time, right? You could have acetylcholine, you could have histamine, you could have some dopamine, some glutamate, some GABA, and all these neurotransmitters are, either excitatory or inhibitory depending on the receptors. And in the synapse, you're likely to have multiple receptors for multiple different neurotransmitters. It's just a big mess, right? To make it even messier, what can happen is, before that signal, that action potential gets to the axon terminal, you can have another neuron interface with that axon. Okay? And if that neuron, let's say, were to release GABA onto that axon, okay, what primarily is GABA? Excitatory, inhibitory? Inhibitory. What that GABA can do is it can actually inactivate calcium channels. What will that do if I inactivate calcium channels? Will it really do anything with polarization? If GABA activates GABA receptors, that'll hyperpolarize. But here we're talking about calcium channels. So if it inactivates calcium channels, what's it going to do? Is it going to cause more calcium to go into the cell or less? Less. And what is calcium important for, going back to here? What does calcium do when it enters the neuron? That wave depolarization comes, causes those voltage-gated calcium channels to open. Calcium enters the axon. What does calcium do?
Releases neurotransmitters.
It enables the neurotransmitters to be released. So even though that impulse, that wave of depolarization gets to the axon terminal, if GABA is being dumped on and inactivating those calcium channels, will the neurotransmitters be released? Not necessarily. So this is what's known as presynaptic inhibition, where I'm actually inhibiting this neuron, but I'm not inhibiting it here. I'm not inhibiting its ability to depolarize, I'm inhibiting its ability to release neurotransmitters. And then likewise, I could also have presynaptic facilitation. Let's say that I dump some serotonin on those calcium channels, and that serotonin causes more calcium channels to open or activates them. What's that going to do then? What's it going to cause this neuron to do? Is it going to release more or less neurotransmitter? More neurotransmitter will be released. So that's presynaptic and facilitation and presynaptic inhibition. Right, so you have a really complex soup of stuff going on here. You guys okay with that? All right, take a break. I'll see you guys in about 10 minutes, and we will push for finalizing this.